Hi, I'm Jolien. I'm a senior scientist with Teiko Bio, and I'd like to present our poster here at AACR 2023 that we did in collaboration with the Parker Institute for Cancer Immunotherapy. This is part of the RADIOHEAD study in which PICE collected patient samples from thousands of cancer patients all over the United States who've been treated with standard of care immunotherapy. The goal of this study is to identify biological features associated with clinical outcomes, such as response or adverse events, from the blood. And PICE reached out to Teiko to help with the immune profiling component of this research. So this is a proof of concept study using, looking specifically at patients with urothelial cancer who were treated with anti-PD-1. And for this study, it's a cohort of 40 patients, 75 PBMC samples. We designed a 44-marker mass cytometry panel that is able to detect all the circulating non-granulocytes of both the adaptive and the innate immune system. We also included about 20 different functional state markers that are able to detect states like activation, suppression, proliferation, migration, etc. And we looked at samples from baseline, before treatments, as well as early treatments, which is a range between day 1 and day 120. So first, we had a look at immune features associated with overall survival using univariate Cox regression analysis. Cell phenotypes means essentially the cell frequencies of the major immune population. Cell states means the functional states of those major immune populations. As we can see, at baseline, we don't see too much, which is in line with what the research shows. We see more interesting data here early on treatments. Interestingly, what we see is that there are several regulatory populations associated with better overall survival, such as regulatory T cells here, as well as TIM3 positive classical monocytes, as well as classical DCs. Conversely, we see activated CD8 immune cell populations associated with worse overall survival, such as the KLRG1 CD8 T naive cells, as well as double positive CD38 HLA-DR effector memory CD8 T cells and central memory CD8 T cells. To get a rough assessment of the impact of these immune populations on overall survival, we did a Kaplan-Meier curve analysis in which we divided patients into two groups. Patients either had above the median frequency for a particular cell population or they had below median frequency of this particular cell population. And we see a similar pattern emerge. Regulatory populations, such as regulatory T cells and TIM3 classical monocytes, are positively associated with overall survival, meaning if they have higher than regular frequencies of the population, they will have better outcomes. While people who have higher than average frequencies of activated CD8 T cell populations, such as these effector memory cells or the KLRG1 positive CD8 T naive cells, have worse overall survival. To have a better understanding of what's going on here, we did an analysis using unsupervised clustering in which we divided patients into 4 groups based on their response status and the presentation of irAE. And we ordered them on the plots based on hypothetical immune activation. So patients who have the least immune activations, non-responders with no irAE, are all the way on the left, and patients who have response and an irAE, most immune activation, are all the way on the right. We did clustering using a lot of different clusters, but we highlight here the regulatory T cells and the NK cells, because we saw that these were perhaps indicative of what would be going on at a grander scale. We see these regulatory T cell populations that are differentiated from each other by CD38, CTLA4, and ICOS expression. TIGIT expression is more variable, indicating that there might be subclusters within these clusters. But what we see here for the regulatory cluster 1, we see that at baseline, we already have higher levels of cluster 1 in the patients who will go on to develop an irAE later during treatments. So they're higher in the non-responders irAE and in the responders with an irAE. That is maintained at early on treatment, which could indicate that these patients maybe have higher baseline status of activation and therefore they have more of these regulatory T cells. What we see conversely with the cluster 2, there's not much differentiation at baseline, but we see specific upregulation of this cluster in the patients who will go on to develop a response to treatments. And it's specifically like a quantitating the change from baseline, we see about a 2-fold change in those responders in this particular cluster. So there is also something going on between the individual T regulatory populations. We also see immune features unique to those patients with response in irAE. For example, we found the peripheral NK cell cluster, we labeled cytotoxic cluster 2, which is differentiated by low expression of KLRG1, high expression of CD161, and low expression of CD57. It is maintained, even maybe upregulated over time in almost all the patient populations, except the patients who will go on to develop a response in irAE. In those patients, it is significantly downregulated. We did gated analysis on these populations to confirm that what our unsupervised clustering algorithm detected is real. We see similar patterns in the regulatory T cells, TIM3 classical monocytes, and our cytolytic CD16 positive NK cells, the ones that we discovered from the unsupervised clustering. Gated analysis also finds novel immune populations that perhaps didn't quite pop during the unsupervised clustering. For example, these non-migrating CD74 CDCs are much higher in frequency, up to 30% higher in those patients who will develop a response as well as an irAE compared to the other cell populations, compared to the other patient populations. So in conclusion, we demonstrate that mass cytometry can be used to detect peripheral blood biomarkers of both response as well as irAE. Some of these immune features might be unique to the different clinical outcomes. Some might be some might overlap between response and irAE. More study is needed to differentiate. For example, doing a more granular analysis of specific time points. We also want to do more analysis on other indications. We're working currently on melanoma and NSCLC of the RADIOHEAD study. And finally, we'd like to thank PICE for their collaboration with us, as well as the JDRF and Helmsley Trust for funding patient collection. Thank you.